Welcome, everyone. Today, we are diving into a fascinating topic in computational biology called high-throughput functional genomics. If you have ever wondered how we go from just reading a static DNA sequence to actually understanding how thousands of genes work together to keep an organism alive, you are in the right place. This field is revolutionizing modern medicine and biology, and by the end of this lesson, you will understand exactly how it works. Now that we have introduced our topic, let us build some intuition using a simple analogy. Imagine you are trying to understand how a massive, complex factory works. If you just walk through and write down a list of all the machines inside, that is like traditional genomics. You have the parts list, but you do not actually know what any of the machines do. Now, imagine you install thousands of cameras to watch every single machine simultaneously. You can see exactly which ones turn on during a rush hour and which ones shut down. That is exactly what high-throughput functional genomics does for a living cell. It takes us from merely knowing the static genetic code to understanding how that code actively operates. Building on that factory analogy, let us break down the actual scientific terms. The word functional means we are looking at dynamic, living processes like gene expression and protein interactions, rather than just reading static DNA sequences. The term high throughput refers to the use of modern automated technologies, such as DNA microarrays and next-generation sequencing. These tools can generate massive amounts of data in a single rapid experiment. Because these experiments produce millions of data points simultaneously, computational biology is absolutely required to process, normalize, and interpret the results. We are bridging the gap between a static sequence and complex biological behavior. So, why does this massive scale of data collection matter in the real world? Without functional genomics, having a fully sequenced human genome is like having a massive instruction manual written in a language we cannot read. In the real world, this technology is heavily used in cancer research, drug discovery, and personalized medicine. For example, by comparing the gene expression of a healthy cell to a cancerous cell using these high-throughput methods, doctors can pinpoint exactly which genes are driving the tumor's growth. This allows medical professionals to prescribe targeted drugs that specifically fix those malfunctioning genes, saving lives instead of relying on generic treatments. Now that we understand the real-world impact, we need a mathematical way to measure these changes. To understand if a gene's function is changing, we must measure its expression level, which is essentially how much RNA it is actively producing. The most fundamental metric here is the fold change. Conceptually, this formula simply divides the new expression level by the old expression level to see how many times larger or smaller it has become. FC stands for the raw fold change, which is a dimensionless ratio. E treatment represents the measured expression level of a specific gene in the treated, stressed, or diseased sample. E control represents the expression level of that exact same gene in the normal, healthy baseline sample. If the treatment value increases, the fold change becomes greater than 1, meaning the gene is upregulated. If it decreases, the fold change is less than 1, meaning it is downregulated. Before we move on to more advanced metrics, let us see this raw fold change formula in action with a concrete example. A researcher is studying how yeast cells respond to extreme environmental heat. We are given that under normal room temperature conditions, which act as our control, gene A produces 400 units of RNA. After exposing the yeast to high heat, our treatment condition, gene A produces 3,200 units of RNA. We are asked to calculate the raw fold change. We chose this formula to find the simple ratio of change. By substituting 3,200 for the treatment and 400 for the control, we divide the two numbers. The final answer is 8. Physically, this means gene A is producing exactly 8 times as much RNA under heat stress, suggesting it plays a critical role in protecting the cell during emergencies. While that raw fold change of 8 was easy to calculate and understand, it creates a major problem for computers. Let us look at why. The raw fold change is mathematically skewed. For instance, an increase in gene expression by a factor of 2 gives a fold change of 2. However, a decrease by a factor of 2 gives a fold change of 0.5. The distance from 1 to 2 is much larger than the distance from 1 to 0.5, even though the biological magnitude of change is exactly the same. This asymmetry makes it very difficult for computational algorithms to plot, filter, and analyze massive genomic datasets without introducing bias. We need a way to balance this scale. To fix this asymmetry we just discussed, computational biologists almost always use the log2 fold change. Conceptually, we apply a logarithmic transformation to make the scale perfectly balanced. In this formula, LFC stands for the log base 2 fold change. We calculate this by taking the base 2 logarithm of the raw fold change ratio we found earlier. By applying this log function, the scale becomes perfectly symmetric around 0. Now, an increase in expression by a factor of 2 becomes an LFC of positive 1, and a decrease by a factor of 2 becomes an LFC of negative 1. If the expression does not change at all, the ratio is exactly 1, and the resulting LFC is exactly 0. This beautiful symmetry is ideal for statistical analysis. Now that we have our new symmetric formula, let us apply it to our previous yeast heat shock example. We are asked to find the log2 fold change for gene A. We already know from our previous calculation that the raw fold change is 8. We choose the log2 formula to make this data ready for a computational pipeline. We take the base 2 logarithm of 8. To solve this, we ask ourselves, what power must we raise the number 2 to in order to get 8? Since 2 raised to the power of 3 equals 8, our result is 3. The final answer is a log2 fold change of positive 3. Physically, this positive value immediately flags the gene as highly upregulated in any computational analysis. Let us look at another critical example, this time focusing on disease. In a healthy lung cell, a known tumor suppressor gene called gene B has a baseline expression level of 1,000 units. In a cancerous lung cell taken from the same patient, the expression drops drastically to just 125 units. We are asked to calculate the raw fold change to quantify this suppression. We use the fold change ratio, dividing the treatment value of 125 by the control value of 1,000. This gives us a decimal of 0.125, which is exactly equivalent to the fraction 1/8. Physically, this confirms that the protective gene's activity has been severely reduced, operating at only a fraction of its normal capacity in the cancer cell. Following up on that severe suppression, we must now convert our result into the log2 fold change format. We are asked to find the LFC for gene B. We take our raw fold change of 0.125, which is 1/8, and apply the base 2 logarithm. We ask, what power 2 must be raised to in order to equal 1/8? Because 2 raised to the power of negative 3 equals 1/8, our result is negative 3. The final answer is an LFC of negative 3. Physically, this negative sign elegantly represents biological downregulation. it immediately flags the gene as heavily suppressed in the computational pipeline, indicating to researchers that the loss of this specific gene might be allowing the cancer to grow. Now that we have calculated these values, let us visualize what this looks like in a computational pipeline. On screen, you are seeing a bar graph representing the log2 fold change for 3 different genes. The horizontal axis represents the specific genes, while the vertical axis shows the LFC values, perfectly symmetric around the 0 line. Look at gene A on the left. Its tall green bar reaches +3, clearly showing it is upregulated. In the middle, gene B has a deep red bar reaching down to -3, instantly identifying it as downregulated. Finally, gene C shows almost no change, hovering right at the 0 line. This diagram teaches us how the logarithmic transformation allows computers to instantly and visually identify significant biological changes across an entire genome. To wrap up our lesson, let us review the most important ideas we have covered. First, high-throughput functional genomics allows us to observe the dynamic behavior of the entire genome simultaneously, moving beyond a static list of DNA letters. Second, while raw fold change gives us the basic ratio of gene expression, it suffers from mathematical asymmetry. Third, by applying the log2 fold change, we create a perfectly symmetric scale that allows computational algorithms to analyze massive datasets without bias. You have now seen how these mathematical concepts directly connect to identifying cancer-causing genes and developing targeted treatments. You have covered the full topic, mastering the first step toward discovering how complex biological networks operate. Great job!